The Bryans present chemistry experiment of wavelengths, max absorbance of red cabbage juice and solutions. The independent variable of our experiment is the pH of the red cabbage juice dye. The dependent variable is wavelengths of max absorbance. The controls of this experiment are pressure, temperature, and volume. Our hypothesis is we believe that when we change the pH, the color will change and wavelengths will vary. First, we create a solution of 5 milliliter base and 5 milliliters of acid. First, we add the base solution to the red cabbage juice, which forms a green color, but over time turns to yellow. Next, we add our acid to our red cabbage dye, which forms a reddish color. Now we fill up the cuvettes with each solution. First, the normal cabbage juice, which is a bluish color, similar to a light indigo. Next, we use the acid solution that was mixed with cabbage juice, which forms a light reddish color. Then lastly is the base solution, which is a yellowish color.
Okay, so there seems to be two places where it peaks off at the most, and the first one, which seems to be ultraviolet, and the highest point there is about 5.809 by 383.21 nanometer, and you can see if you get the text in there. Yep. And then there's another peak, and it appears that this one's in the visible light spectrum, the other one's in ultraviolet, near the ultraviolet. And
As you can see, the base solution of red cabbage juice is being absorbed by bluish and purplish colors, which describes them as yellowish tints. And the absorbance peaks at about 3.0, and the wavelength at this point is about 394.46 nanometers.
Okay, so here's the acid solution, and it appears that the wavelengths absorbed is all near the green in the visible spectrum, and its absorbance max is about 2.1131 with a wavelength of 516.45 nanometers, and it appears that none of the red is absorbed, so that's why the colors turn out red.
A random error that may have occurred in our lab was that because we may have cleaned off the pH probes, so it might have contaminated the other solution, which causes them to change color. For example, the base, which turned from a green to a yellow. A systematic error that may have occurred in our lab was that we should have used aluminum foil to cover up the beakers, because the air may have contaminated the base solution, which caused the base to change from a greenish to yellowish color. Practical application of this lab is to show that acids and bases can affect the color of the cabbage juice, and that if the cabbage juice turns into a reddish color, we know that it's slightly acidic, and if the red cabbage juice is like yellowish greenish, then we know it's kind of basic.